Today I'm going to talk to you about mutations. This is one of two lectures that are going to cover what leads to the variation in the phage genomes that you're working with. You know that there is a fair amount of genetic variation within these genomes because every phage that you have looked at has got a difference in sequence. They're not completely 100% the same. And that has led, of course, to the variation that leads not only to phage that can grow on the different mycobacteria or other bacterial hosts, but created the clusters that we look at. So the phage, or the mutations that occur, can occur anywhere in the DNA that I'm going to talk about. And they can have negative effects, positive effects, or be neutral. There are a large number of mechanisms by which mutations occur, and I'm not going to cover all of these. Some of these are replication errors. I'm going to tell you a little bit about chemical instability. UV light, of course, you're all familiar with because it leads to things like skin cancer. Error-prone repair of the damage, things that stick into the DNA, like ethidium bromide, can cause mutations and other possibilities, including errors during genetic recombination. And I'll talk a little bit about some of that because that's also going to be relevant to what happens in our discussion next week. So mutations can occur during replication. Normally, so I have this circle represent the DNA polymerase, and it's sitting here on a template, and it's reading this template 3' to 5', it's synthesizing 5' to 3', and it's going to add, it's going to add an A at this position, right, just like that. And if the polymerase is going to continue, then it would add a G and so on. But polymerases are only human, and so they sometimes make mistakes. And so instead of putting in an A, it happens to put in a G. It actually adds a G covalently onto this T. Now, the G does not pair very well with the T, but it can pair a little bit, and maybe it'll stick there just long enough for the polymerase to add another G. Now we have a problem because we have a mismatch here between a G and a T. And I'll talk to you in a little bit about how the cell deals with some of these. But by and large, we've now created a mutation on one of these strands. Fortunately, this may happen fairly frequently, but fortunately, the DNA polymerase has got a way of handling this problem in that has a backspace key. So when an incorrect base gets added, there's an exonuclease that will swing into position. So the first thing that happens is it gets displaced because it's not held there very well by hydrogen bonding. The exonuclease will swing into place, will clip the bond between the G and the preceding the nucleotide, and then everybody goes back to place and the G goes off, and now we have the ability to add a correct A at this point, followed by a G, and so on. So the A gets added, and just to show you this is a more correct type structure, here's where the mismatch has been added. Here's the A exonuclease active site. The DNA swings out of the way, the exonuclease cuts off the extra base, and then replication proceeds. So chemical instability is another way in which DNA gets mutations, and this is something that happens a lot. This actually happens a lot in our body, several hundred times a day. Fortunately, we have mechanisms that repair these problems. And what I'm going to show you here is a mutation that's actually caused by water. So the water comes in, and in your organic chemistry, you'll learn about something called a nucleophilic attack, that this oxygen will attack this bond, breaking the nitrogen bond, and coming in here and ultimately leading to this structure. So this molecule is a cytosine. You can tell that by C. Hopefully you will actually recognize it as a cytosine. This molecule is a uracil. As you can tell from the U, also I hope you recognize the structure. So now we've made a mutation from a cytosine to a uracil. Cytosine pairs with G, uracil will pair with an A. Now uracil does not normally occur in the DNA, and so the cell has a way of recognizing this and will get rid of this. But if this is still there when DNA replication goes on, then this uracil will specify an A on the other strand. The problem becomes even worse in DNA such as our own, because in humans and other eukaryotes, there's a significant fraction of the cytosines that contain a methylation on this 5' position. So in our DNA, about 3% of our Cs actually have this modification. Now if this water comes in, cleaves off this nitrogen, So that goes off. The hydrogen or the water comes in. The oxygen remains here. Now this structure, instead of being a uracil, is a thymine. And this is exactly what's supposed to be in the DNA. So now we have a problem because we'll have this thymine on one strand and we'll have a G on another strand. Okay, so that's similar to what I showed you with the error in replication. Another way in which DNA can get damaged is UV light. And this, as I said, you'll recognize this sort of thing because of the problem of people getting too much sun exposure leading to damage to their DNA. So what happens with UV light is the energy now takes the bonds that each thymine, if you have two thymines next to each other, it'll break the bonds along this backbone side here, and will create new bonds between these two structures. And what that does is that instead of DNA having a displacement of about 36 degrees as it spirals on a helix, these two things are now bound in such a way that they're directly stacked one on top of each other. And they are not actually in a nice order in the helix. So we can see that in a model of the DNA. So here's the bases, and the bases along here you can see come right out of the backbone on a flat structure. Now this one is actually a sort of poor representation. There should be a bond here and there should be a bond here to create the cyclobutane ring that are between two thymines, right? And then if we look on the helix side of things, we see that the bases are not coming out flat, but they're coming out at an angle relative to each other. So that if we were to put in the other strand, you can see that they don't match up perfectly. They're coming up at an angle relative and down at an angle relative to the bases on the other side. So there's mismatch, and this disrupts replication when the cell tries to replicate the DNA. Another way in which we can get errors or mutations is errors during recombination. So what I've shown here is a diagram of a chromosome, essentially two chromosomes, such as they might be during meiosis, but the same thing can happen in prokaryotic cells when you have two sequences that are related to each other coming in pairing. So I've got three genes shown here, and they're color-coded. In this one, they're the same alleles on both of them. This one's got a purple and a pink allele, and this one's got a blue and a cyan or teal allele, I guess cyan allele. And if we have recombination in this point between these two, then what would happen is that we would create a chromosome that would start up here, go through the purple allele, and down to the cyan allele. And we have another chromosome that would be created by coming through these two genes and then go up and pick up this dark blue allele. And so the result of that in recombination would simply be that we've switched these alleles. So we have this chromosome now created by going from here to here, and we have this chromosome created from here to here, but all the genes are in the same order. All we've done is swapped alleles. The problem comes is if there happens to be a sequence here that looks like a sequence here, and we get a recombination between this site and this site. Now what happens is we would come through this gene to this gene, but then we would come over to here through this particular gene, now the pink allele to the light blue allele, and then we go through here up into this one. That would create recombination products that would look like this, one of which is this one. Okay, so what has happened here is this gene has been completely deleted, and when we would have this one, and now what would happen is we have two copies of the same gene. So that's a recombination error that has led to different structures. The deletion, if this happens to be an essential gene, would be fatal. Okay? If we have genes in which the copy number is important, then we would have a duplication, and that could also disrupt development, or it would allow two genes which could now start separating in evolution one from the other as they create individual mutations. So organisms are protected from damage to DNA by several mechanisms, one of which is called base excision repair, in which just the base is removed from the backbone and replaced. Another which is called nucleotide excision repair, in which the backbone is actually broken and some of the nucleotides removed and then repaired. And then information retrieval, which is getting information from another chromosome. So nucleotide excision or base excision repair occurs, for example, when we form this uracil. There's an enzyme that recognizes it is the uracil, cuts it off, leaves a single position where the sugar does not have a base added. There are two more enzymes that come in and cleave that out, and then DNA polymerase will fill this little gap in. So now we would get, and it would use, of course, the information on the other strand to insert the correct base. Nucleotide excision repair is similar, except it's a little bit more complex. We have a mechanism, and I'm not going to go through the details of this, where it recognizes a mismatch. So this could be that TG mismatch that occurs. And what it does is the system will take and create a break in the DNA, will degrade the DNA, then fill the DNA, that empty spot. So it degrades past the mismatch, fills in the empty spot, and then it's now completely repaired. Now, there's some danger here because in some cases, the base that got changed is the one that gets retained and not the one that was there originally. And that can lead to a mutation. But in most cases, actually, there's a system set up so the changed base is the one that gets preferentially removed. So the final repair mechanism I'm going to talk about fairly quickly is what happens when you have a double-strand break in the DNA. Now, there's a process I'm not going to talk about at the moment, which is called non-homologous end joining, which puts these two ends together. But unfortunately, the process requires damage to the DNA, so we lose some bases at this point. So that creates a place where we have one or more bases removed in the DNA, and now you've lost some information. And that's critical if it's part of a protein coding region. But normally what happens if we have a break is the cell tries to grab information from a closely related, if it's in a eukaryotic cell, it's the chromosome that we all carry. If it's a prokaryotic cell, it might be grabbing from some other or recently copied genome, or it might be from grabbing from some unrelated sequence, for example, a plasmid or a phage. So the process is that there's degradation of the ends, then it creates a single-stranded end, which then invades into the helix of this other piece of DNA, copies that information. The displaced strand is also used for copying information, and the resulting information up here is specified by the DNA that is down here. So if this DNA is identical to this DNA, then there's no mutation that has been created. But if this DNA is different, now we have inserted a new sequence from this other piece of DNA. So that's something to think about when you think about the changes that have occurred in the phage genome. Some mutations lead to single nucleotide changes, and then these could be silent mutations if they do not change the amino acid. They could be missense mutations if they do change the amino acid, or they could be nonsense mutations if they lead to the incorporation of a termination codon. All of which have got some, or these two of which have got some consequences. Obviously, it could be a lot of consequences. Silent mutations will have very little consequences, except as it might change the codon that's being used. And you know about codon bias and the use of codon bias. If we have something that leads to incorporation of DNA, that can lead to a frame shift, or if we have an insertion or deletion, we can get frame shifts in the coding region. You know the problems that could lead to. You could lose a gene, you can gain a gene. So the other point I'll just make out is that new mutations become permanent when replication occurs before repair, because we do have these elegant repair systems. So if we have damage to DNA where we get or opposite this A, we now have a G instead of the T that's supposed to be there. Then if this gets repaired, it just simply goes back to here. And then during replication, it will go through this normal process, leading to normal products. But if replication takes place before repair, then we start off with this in the template. Upon replication, then we wind up with this product, which gives us back our normal AT base pair. But because there's a G on this strand, we wind up now with a GC base pair. So that's the situation that I was talking about when you had an error by DNA replication and if it does not get repaired. So I'm just going to conclude with some things for you to think about before class on Friday. One is, where in the genome do mutations take place? And if they are localized, when you look at them, when you find them in a localized place, why are they localized? How many of the mechanisms that I've talked about may play a role in phage mutagenesis? How is replication of the phage the same as the host, and so therefore how is it different, and that will impact repair? Repair often requires another source of DNA to provide a template. How do the bacteria do that? And I've already given you a hint on that one. And what are the possible sources of genetic material to provide a template for repair and variation? And I've already given you a hint on that one. And with that, I will stop.